Hello everyone, my name is Jayaram Kancherla. I work on developing interactive visualization and exploratory tools for genomic datasets. This includes epigenetic data, metagenomic data, and my topic for today, which is single-cell RNA-seq data. A motivation for this work comes from understanding how analysts analyze and explore single-cell RNA-seq data, walking through some of the steps as shown in the workflow on the right. Once the reads are generated from a sequencing machine, they're run through standard pre-processing pipelines to generate a count matrix. The analyst then performs preliminary QC checks to filter out cells that did not meet certain expectations and generates a filtered count matrix. Now, an obvious next step is to perform clustering by using either supervised or semi-supervised methods. Depending on what methods they choose, they either specify a clustering resolution if they use graph or community-based methods, or a number of clusters K, as in k-means based approaches. Once they identify these clusters, they can then do further analysis downstream and identifying marker genes or differentially expressed genes, or perform trajectory analysis and so on. An important step in the downstream analysis is to first identify different cell populations or clusters, and depending on the use case, also determine what these cell types are based on known marker genes. Usually, the analyst performs this analysis several times, trying out different resolutions if they're using graph-based approaches, or different values of K if they're using k-means based approaches, to finally get a good set of clusters to use. We find this step in the analysis to be highly iterative and explorative process, and we think this is where single-cell TreeVis can help. SC TreeVis is an interactive exploratory tool for visual analysis of single-cell clusters at different resolutions. If you're looking at the figure on the right, we use the facet zoom technique to allow users navigate the hierarchy of clusters at different resolutions. In addition to navigation, the facet zoom also allows users to aggregate and remove clusters across resolutions, and I'll show a few examples of these in the next couple of slides. There are also multiple visualizations to support an analysis. This includes visualizing lower dimensionality plots or embeddings, for example, t-SNEs, UMAPs, or PCA, and also visualizing the count matrix itself. We provide heat maps to visualize an aggregated count matrix to the cluster level, or box plots and scatter plots to explore the expression of a single gene across clusters. When the application is first initialized, it uses the default resolution. It sets a default resolution, and from the facet zoom in the figure, that is currently at cluster resolution 4, and all visualizations that you add to the workspace, you use this as the cluster grouping. So the data in the heat map is the count matrix aggregated to cluster resolution 4, and the heat map also provides a way of dynamically clustering across genes or cells or even both. All the color codings that you see on the clusters also use the cluster resolution 4, and the blue cell and all the cells are colored using this coding. Brushing is also implemented to quickly highlight cells across all the visualizations in a workspace. So for example, if you hover or brush over a node in the facet zoom, it immediately highlights all the cells that belong to that cluster, both in the heat maps and across all the lower dimensionality plots. A critical component in exploring clusters at different resolutions is the facet zoom technique we borrowed from information visualization research. If you're familiar with the older iPods and how they work, this is very similar to how users can find songs in the catalog. So you first start with all songs, and then as you drill down the hierarchy, depending on what the first initial characters of the song are, you get to filter down the list of songs in your iPod. In addition to navigation, the facet zoom also provides you a context of where you are in the navigation in the tree itself, or the hierarchy, and that's shown by the zoom or the lower levels on the sides. We use the exact same technique to allow users to explore hierarchies, single cell hierarchies across resolutions, but we also allow a few more interactions on the nodes of the facet zoom. For example, let's say you're exploring a data set, and you notice in one of the lower dimensionality embeddings, PCA in this case in the figure, the clusters that are circled, the orange, red, and green clusters and cells are not clearly well separated. So you as an analyst or the user that is trying to analyze this data set might want to group them as a single cluster rather than 3 separate clusters. You may notice that the cluster resolution is 4, but if you want to group them all together, we go to its parent at cluster 2C1, which is at cluster resolution 2, and aggregate the cluster. This step is very useful and important in the application because it allows users to define clusters across resolutions that is not possible when you're using traditional analysis methods. Once the cluster selection is made, it updates all the other visualizations in the workspace. So a new aggregated count matrix is generated on the fly dynamically, and the heat map is updated. And all the color codings across all the lower dimensionality plots and any other visualizations that you add to a workspace are updated based on this new change in the cluster order from the facet zoom. Similarly, if a user is not interested in a cluster, he can choose to remove this cluster from the analysis. This removes the counts from the heat map, but all the cells are grayed out in the lower dimensionality plots. So this still provides you an overview of where the cells are in the embedding space, but not be as prominent as the other ones. The application supports multiple ways. Not everything is shown here to explore expression data or the results of an analysis. This includes bar charts, violin plots, scatter plots for exploring expression of a single gene across all the cells, and many of the other visualizations I have shown before. Depending on what type of data is available in the dataset, a lot of flexibility in customizing how to add visualizations to a workspace. For example, if you have the topmost variable genes for a dataset, you can choose to add a heat map for these for only the topmost variable genes rather than all the genes. You can also look at the expression of marker genes across all cell types as box plots, bar plots, and violin plots. And I've shown through some of the interactions before, all the charts are linked to each other, so brushing on one will immediately highlight that cluster across all the other cells, all the other visualizations. And making any changes on the facet zoom will immediately update all the other visualizations on the application. We had to do a little bit of engineering into how we quickly visualize large matrices or large render expression data in heat map. There's a possibility that the user, as they drill down the facet zoom, the hierarchy in the facet zoom, they may want to look at the entire data set at the lowest resolution possible. So we use this technique that is commonly used in video games called pixel manipulation rather than higher level canvas APIs to render large matrices very quickly and enable interactive visualization of single cell expression data. In addition to rendering, we also had to optimize aggregating large count matrices as the user makes many of these selections on the facet zoom. This is possible by taking advantage of existing biocontent data structures that represent single cell RNA-seq data. So this includes both SURAT and single cell experiment objects. Using these standardized data structures allows us to optimize and quickly perform these aggregation operations. The package is currently available on GitHub and is currently under review at Bioconductor. One of the unique features of the package is once you initialize the application using either a SURAT or a single cell experiment object, and you make these selections of clusters on the application or any changes, they can be brought back from the application back to the R object and can be used in any of the further downstream analysis. So you can use the cluster ordering, cluster grouping that you've chosen across clusters, across resolutions from the app, and then perform differential expression or marker gene analysis downstream. With that said, I want to thank all our collaborators, especially Stephanie Hicks, my advisor Hector Corrada Bravo, and Kasi Zenith, who is a graduate student in the lab, for all their continuous help, support, and feedback while we are developing the application and the R package itself. So please check out our application and give us any feedback on how we can improve the interactions or support your analysis of single cell datasets. Thank you.